you have not prior cleared that, at this point, put the phones up. You do not want to see anybody's phone up except anybody who has specific permission already. I think it's only like one or two that I've heard. So phones should be up, ringers should be off. You guys know where the restrooms are. If at any point today you need to go, you don't need to ask permission. Just quietly out there in the hall, not in the hall, but in the corner out there, is where you're going to find the men's and ladies' rooms. I think you know that. So we are very happy to have with us today Dr. John McDowell from Virginia Tech. He has been at Tech for 17 years, working in plant biology, primarily for looking for ways to increase agricultural production and other aspects of plant genetics, I'm guessing. This is part of one of the real neat things about this project is we are connected to a lot of real work and research that's going out there. So I'm going to turn it over to Dr. McDowell to explain a little bit about Arabidopsis. Please give him a warm welcome.
Thanks, guys. I'm really happy to be here, and I'm looking forward to working with y'all this semester. Let's see. Is there a pointer on any of this?
Oh, you need one.
Got it.
Oh, yeah.
That's good enough. Okay. Okay, so what I'm going to do is talk to you guys for a little while about the research that goes on in my group and how it kind of fits into the bigger picture of global challenges that we're trying to address. And then we'll talk more about the specific mutation that you guys are going to be helping us understand, how it affects plant productivity and how that will then tie back into our grand plans to boost agricultural productivity. At the heart of, my lab's research is an intense desire to make food production more secure and more efficient. Agriculture is pretty hard on the environment. You guys have any idea how much I'm getting a high sign from back. Is this? Oh, right on.
Be 1 second. I gotta get you some batteries.
No worries. I'll try to talk loud in the meantime. Okay, and actually, you guys are going to talk a little bit. How much of the world's fresh water do you think is devoted to agriculture? Yes, it's 80%. That's pretty close. It's a little bit high, but yeah, it takes a lot of water. What about greenhouse gases? What percent of the world's greenhouse gases actually come from processes associated with food production? 5%. Got it right, boss. 2, that's a little bit high, but most estimates are in the neighborhood of 40%. If you think about it from that standpoint, food production is really bad for the environment in terms of water security and global warming. So we want to make it more efficient. And one of the big drains on agricultural productivity is diseases like the one that's shown in this picture. You guys are pretty smart. Can you guess what plant we have here? It's not Arabidopsis. Cannabis? No. So that's a good guess. It's a really important food source.
Tomatoes.
I heard it.
Tomatoes.
Close. Potatoes. Yes. Right. Potatoes. Very good. Okay. So if y'all are so smart, tell me what's going on here. It's a blight. Yes, that is exactly right. And the name of this disease. Okay, am I mic'd up now? Is that better? Excellent. Okay. So the name of the disease is late blight. So that's the simple name. The fancy Linnaean binomial name is Phytophthora infestans. Anyone know what Phytophthora stands for? It's a Greek word. Anyone want to guess? What about phyto? Does that mean anything to you guys? What does that suggest? What? Plant, yes, very good. Okay, so phyto, it has something to do with plant. What might the other part be just based on what you see here? Pardon? So the first part is plant, and the whole word is Phytophthora. Exactly, yeah. So the Phytophthora is actually Greek for plant destroyer. You guys can see where the disease gets its name. It's really laying waste to this potato field. Now, this particular disease has a lot of historical significance, and I'm sure you guys have all heard of it before, even if you don't realize it now. Any ideas on that? The Great Potato Famine. Yeah, Phytophthora infestans was the pathogen that caused potato famine in Ireland, which is generally thought to have killed between 1 and 2 million, and also led to the immigration of about that number of people out of Ireland. I know people who trace their residency in the United States to their ancestors' immigration from Ireland to the U.S. as a direct result of this disease. Okay, now some more not so fun facts. This disease is still a big problem today. It's estimated to cause about $6 billion, and that's billion with a B, $6 billion in crop loss. And just to make that number a little real, if you could buy potatoes at current market value, so if you had that $6 billion to buy potatoes today, you can meet the caloric needs of the entire world for about 2.5 billion. So that's a lot of food that is lost to this one disease. Now, in the next slide, I'm showing several diseases that are caused by relatives of the late blight pathogen. So these are all different species of the same genus, so they're very close cousins. So they're diseases of crucifers that are really healthy but taste really bad, like cabbage, broccoli, so on. There's a really bad pathogen of soybean. And then these Phytophthora species can also cause a lot of damage in natural environments as well as on the farm. This particular pathogen is, like I said, a close relative of Phytophthora infestans, but it goes to a completely different set of species. It infects a large number of hardwood trees. Amongst those are some of the keystone species of oak that are really important contributors to the ecology of forests on the West Coast. So these are all currently being severely threatened by this particular pathogen. Now, so far, the distribution of this pathogen in the U.S. has been confined to the West Coast, but it's going to spread east. It's not a matter of if, it's just a question of when it eventually happens. It's come close a couple of times due to accidental transport on nursery stock. In those instances, they've been able to maintain the transport, but sooner or later, it's going to escape and it will come here and probably cause a lot of damage on the keystone species that you guys see every day on Facebook. Okay, so all of this is to say that plant diseases have a lot of societal importance. So that's why me and the people in my lab group are so interested in understanding how pathogens like this cause disease and using that knowledge to perhaps come up with some new solutions to disease control. Now, I'm going to talk a little bit about what we know about how pathogens like this cause disease in plants. And the metaphor that I want you guys to have in mind is to think about plant pathogens like this as bioterrorists. So the reason that I like that metaphor is that plants actually have a really efficient immune system that works in many of the same ways that our own immune system works. There are proteins in plant cells that are dedicated to surveillance against pathogens like these Phytophthora species. You think of these molecular sentries. And, these sentries, their job is to profile molecular signals that flow around cells for evidence of potential invasion. And so, just like a Homeland Security agent, that's working at an airport, if they see some signal that alerts them that danger is in the vicinity, well, then they hit an alarm button, and all of these defense mechanisms are activated. Okay? The system usually works pretty well, except when it doesn't. And in these cases that I've described to you, the pathogen has figured out ways to sneak around the system or to nullify its action in some way. Okay, so we refer to these as successful pathogens in the sense that they've been able to infiltrate the plant homeland security system, grow inside the plant, and then begin taking away resources from plant cells to fuel their own growth, and eventually cause destruction of the host, and then we move on to the next victim. So one of the big questions then in my field that guys like me are working really hard on is to understand the genes and proteins that enable plants to do that sort of thing. All right, I've already talked about those two pathogens. Let's talk a little bit about what's currently being done to control diseases like this. So the two main control measures kind of fall under two categories. One that you guys are probably most familiar with is simply the use of pesticides. There are fungicides that are pretty effective against pathogens like Phytophthora, but they come with problems. Okay, and those are all kind of illustrated in this image here. Okay, so here we've got a farmer riding a tractor. Okay, he's spending time that could be better spent on other things to take care of his crop. In the case of that potato pathogen that I was talking about earlier, in a bad disease here, you typically need to do 15 to 20 treatments over the course of the growing season. So it's really intensive in terms of time. It's also intensive in terms of expense. These chemistries cost money to apply. And then they too also have bad side effects on the environment. I don't know if you guys can see this in the back, but it looks like the grower here has his son riding shotgun. The grower himself, he's wearing a space suit and a respirator. No respirator on the kid, but it looks like he has a football helmet on, so I guess he falls off the back of the tractor. Anyway, the point of all of this is just to illustrate that these chemical controls can be really effective, but they carry costs of their own. And another big downfall of this sort of approach is that if you keep applying the chemicals time after time to the same field, you're putting a lot of pressure on the pathogen population to evolve ways to resist the effects of the chemistry. Sure enough, what's been shown time and time again is that pathogens like this Phytophthora species are very good at evolving resistance to repeated chemical treatment. So, this sort of approach can work well in the short term, but not in the long term. So we need new solutions. And so then one of the, like I said, one of the major goals for the research that my group does, and a lot of other groups do on a worldwide basis, is to understand how pathogens like this cause disease, and then to turn that basic understanding into some new strategies for disease control. And I'll talk at the end about how we're thinking about modifying plant genomes to make the plants themselves more resistant, and how you guys are going to help us gather some data to figure out whether the approach that we have in mind is feasible or not. So I'm going to talk a little bit more about that, but first, do y'all have any questions about what I've talked about so far? You're not? Okay, so the particular diseases that I've talked about are not autoimmune. They're kind of the opposite. These pathogens are really, really good at turning the plant's immune system completely off. But there are different types of plant diseases that do resemble autoimmune in the sense that an inflammatory response, if you So that can happen. Good question. Any others? All right. So I want to talk about Arabidopsis for a couple of minutes. So you guys probably know that Arabidopsis is not a crop. It's very small. It has absolutely no commercial value at all. But my group and a lot of other groups like Arabidopsis a lot as a model system because of the ease and simplicity with which we're able to grow it and do experiments. And you guys are going to experience this firsthand. And so it's generally considered as a model organism. And so the metaphor that I like to use for why we work on model organisms is, okay, imagine a scenario in which you're an extraterrestrial, you land on Earth, you see these things with internal combustion engines buzzing around, and you want to figure out how they work. Is it better to study something like this or to study something like that if all you want to do is learn the basic principles of an internal combustion engine? Well, the answer is obvious, right? It makes more sense to start with a simple system first and then apply what you learned to the more complex systems. And so that then is exactly the rationale why guys like me work with Arabidopsis to better understand plants, or why people work with fruit flies or mice instead of human beings if they want to human. So really, it's a question of simplicity and logistics. Okay, so like I said, Arabidopsis doesn't have any commercial value itself. It's a member to the mustard family, so it's related to things like broccoli, cabbage, cauliflower, Brussels sprouts, vegetables that are healthy but taste really bad. But itself, I've tasted it before. It's bitter because it has all of these sulfur compounds, just like broccoli, cabbage, cauliflower, etc. do. But it, again, it has absolutely no economic value. But it's a great organism for genetics and genomics. And one big reason for that is because of its size. Just to illustrate that, okay, here's a can of Red Bull. What you guys will see in about 8 weeks is that the height of this full-grown Arabidopsis plant is about the same as a can of Red Bull. So it really is quite small. And so what that enables guys like me to do is to conduct really large-scale genetic experiments inside a growth chamber inside of my lab building. We can do these year-round instead of having to take over a whole field at Virginia Tech and be limited to one growing season. Do the sorts of things that we want to do. So convenience is big. The life cycle is also short. You guys will see this organism go from a seed to a seed-producing plant in about 6 or 7 weeks. And that, that's super fast compared to any crop. And another really nice aspect of this is that it's really easy to modify the genome. I could take any of you guys and one afternoon teach you how to insert a foreign gene into an Arabidopsis plant, and I guarantee it would work the first time you tried it, and every other time you tried it after that. And that really is a feature that distinguishes Arabidopsis from almost any other plant. And it's a major experimental advantage. Okay, now in this slide, what I'm doing is introducing the pathogen reviews for a lot of our research. Now, I was talking about these Phytophthora species a few minutes ago and how bad they are for agriculture. Well, as it turns out, Arabidopsis is really efficient at resisting almost all of those Phytophthora species. There's actually a species that's very closely related to Phytophthora that causes disease on Arabidopsis populations out in the real world. And so the name of the disease is downy mildew. And that comes from the structures that you see here. So these are structures that were produced at the end of the pathogen's life cycle, where it's grown inside the plant and then grown back out to the outside and made these little tree-like things that produce spores that grow onto other plants. So in a really heavily infected plant, you get this kind of downy appearance, and that's where the name of the disease comes from. Now, the actual name of the organism that causes this disease is Hyloperinospora Arabidopsis. You know, and every time I say that with this slide, I wish I had a little bouncing ball that goes to each syllable. It's really long and complicated. What I'd like you guys to remember is our nickname for this organism, which is simply HPA. Okay? So we've got the plant, which is good, versus the HPA pathogen, which is bad. Now, that slide shows you kind of what happens on the outside of a leaf at the end of the pathogen's infection cycle. This slide shows what's going on the inside. Okay, so this is a picture that we took with a microscope using plant material that was stained with a stain that specifically reveals the pathogen's infection structures. You can see that as these little squiggly lines. So what's going on here is that we've inoculated spores from the pathogen, like little round microscopic balls, onto the surface of the leaf. When those spores sense that they're in a place where they can cause disease, they attach themselves to the surface of the plant leaf. And then they produce these thread-like structures, and we call these hyphae. And these structures penetrate from the outside of the plant leaf to the inside, and then they begin growing in the space in between individual plant cells. So they just kind of snake along like that, and occasionally they branch. And that leads to an infection that looks something like this. Now, the other thing that they do as they're growing in these spaces is to actually penetrate plant cells. And here's a picture that illustrates what I'm talking about. Okay, so you can think of this green blob as kind of a schematic of a plant cell. And the other thing that the picture shows is these pathogen structures here, which we call haustoria. So what's going on here is that the pathogen has penetrated the cell wall that exists around all plant cells. And it's made this protuberance that goes inside the cell and just kind of forces its way in. We think this is really where the action is. This is the location where the pathogen is sucking out the sugars, the amino acids, the water, and the other nutrients that it needs to grow and proliferate. And then at the same time, it's actually sending its own proteins to the inside of plant cells to sabotage the immune system and reprogram the plant cell in such a way that it's more accommodating for invasion. So there's a lot going on here, and a lot of groups besides myself are interested in understanding the details of that. Now, another really interesting aspect of this pathogen is that it is an obligate biotroph. Okay, what does that mean? Well, obligate simply means that the pathogen can't grow in any other environment besides a living plant host. So we can't grow it on a petri plate or culture it in any way. You know, the only way that we can keep it going is to collect spores, spray them onto a fresh plant, and go on from there. Another aspect is it's a so-called biotroph. What that means is that it can't obtain nutrients from dead plant cells. It has to do all of these things while keeping the plant cells alive so that they can serve as nutrient sources.
We're having a hard time hearing.
Really?
Now we can hear you. Hold it up there.
Okay.
Sorry.
So I'll practically have to slaughter into the mic. That's okay. Hopefully you guys have a disinfection protocol. I'm serious. I'm recovering from pneumonia. Handle this with care. Okay. So, right, everyone shrinks back. Okay, so this is kind of a list of the challenges that HPA and the pathogens that it's related to have to overcome to be able to cause infection in plants. So potentially, you know, this pathogen does have some vulnerability. Before we get into this, do you guys have any questions about what I've talked about? It's a really good question. It belongs to a group of pathogens that are called oomycetes. This is a group of pathogens that looks a lot like fungi, you know, like molds and things like that. But this particular group of pathogens is actually related to things like kelp or roundhouse. It's a eukaryote, right? It does all of the things that every eukaryote does. Okay. So a lot of the work that we do on this pathogen was based on what we learned from sequencing its genome. You guys have all heard of the Human Genome Project, right? Well, we had an HPA genome project a few years ago. Learned a lot from that. One of the most cool things that we've learned is that, and a lot of the species that it's related to have lost the ability to take up nitrate and sulfate from the environment. Why is that interesting? Well, a lot of, you know, so-called autotrophic organisms depend on their ability to take up nitrate and sulfate because they're the best sources of the element nitrogen and sulfur that then serve as building blocks for amino acids and other important things that living organisms need to grow. So we saw that immediately as an insight into a potential vulnerability that we might be able to use to attack the pathogen. And so, okay, the question then is, well, if this pathogen can't get nitrogen and sulfur in an inorganic form from the environment, then how is it getting these elements? And so our hypothesis then is that a major aspect of the pathogen's life cycle is to steal these elements from the host plant in organic, like amino acids or small peptides. And I'm going to talk a little bit more later about how plants make and transport amino acids, because that's kind of a key part of how we think about this meeting that you guys are going to be doing your work with. But, you know, the important thing that I want to convey here is the hypotheses that guide the research that relates to the mutant that you guys are going to be working on. So our first hypothesis is that host-synthesized amino acids and peptides are an important source of nutrition. Okay? This is a big strategy through which this particular pathogen is getting the things that it needs to be able to grow and proliferate. Now, another key part of our thinking is that we believe that HPA is somehow manipulating the plant proteins that synthesize and transport these amino acids. So to put it another way, it's reprogramming the plant cell to serve as a sink or a food factory in such a way that the amino acids and whatnot are being diverted to the sites at which the pathogen is establishing these structures. You guys have any questions about those aspects? This is really a key aspect of why we're doing the particular research that we're doing that involves this mutant you guys are going to be working on. So take a minute and think about it. And if there's anything that's mystifying about that, then let me know. Yeah.
So does it, at the end, does it shut down the plant because we do things where they need to go and we do things?
Exactly. Yeah, and I'll show that schematically in a couple of minutes, but that is exactly how we're thinking about it. So the question was whether the pathogen is actually shutting down some of the plant systems through its strategy to divert nutrients from, you know, where they're useful for the pathogen to where where they're not useful to the pathogen to where they are useful. And yeah, we believe that that's exactly what's going on. An important aspect to consider with these hypotheses is that what the pathogen is actually doing is taking mechanisms that the plant itself uses for amino acid biosynthesis and transport and simply reprogramming them so that they work more efficiently for the pathogen's benefit instead of the plant. I'll come back to that concept in a minute, but keep it in your mind for now. Okay, so what do we do to test this hypothesis? Because, right, when you formulate a hypothesis, it's useless unless it leads to experiments that you can do to test whether it's actually true or not. And one of the first things that we did then was to look at genes in Arabidopsis, which we know play a role in biosynthesis or transport of amino acids. So if our hypothesis is correct, what one would predict is that the pathogen is capable of reprogramming the activity of the genes that have to do with amino acid biosynthesis or transport. So, okay, so time's kind of short. I can't go through the details of the data that I'm showing up here. But what this provides is kind of an overview of all of the genes in Arabidopsis that are involved in amino acid transport and synthesis. And so what these red lines are telling us is that for the corresponding gene in a diseased plant, the activity of a lot of these genes is changed compared to plants that are capable of resisting this particular strain of the pathogen. So that observation then is consistent with the idea that the pathogen is somehow manipulating these processes inside the plant. We took that as good news. So then the next step was to get mutant alleles of a lot of the genes that the pathogen is reprogramming, and then to actually take those individual mutant plants and infect them with the pathogen. Okay, so here's a thought exercise for you guys. If there's a plant gene that's working for the pathogen's benefit, if you test a plant in which one of those genes has been inactivated by mutation, what effect do you think that might have on the pathogen's ability to cause disease? Bingo, yes. And so its growth would be decreased. So that was exactly the effect that we were looking for in that next set of experiments. And for some of the genes that we tested, including the gene that you guys are going to be doing your experiments on, that's exactly what we saw. So what I'm showing you in this graph is the amount of pathogen growth in a wild-type Arabidopsis plant compared to plants that have mutations in individual amino acid transporter proteins. What you can hopefully see is that in all four of these, there's a reduction in pathogen growth. What we've subsequently learned is that for some of these four genes, when we put the two mutations together in a single plant, growth is reduced even further. So what that is telling us then is that these genes are necessary for the pathogen to get across the full amount of genes. That's exactly the outcome that we were looking for this sort of experiment. So we were pretty stoked when we got this result. Now, all four of these genes encode proteins that function in transport of amino acids. So I want to explain that a little more fully so that you guys can think about what the function of these sorts of genes are in a normal plant that's not being infected by a pathogen. Okay, so you guys know very well that amino acids are important for growth and cellular function and all of that. They're the building blocks of proteins. In plant cells, they also contribute carbon that's very important for cell growth and function. And they're also the building block for secondary metabolites that carry out a lot of important plant functions. Now, a really important aspect for you guys to be aware of is that there's specialization in terms of which plant organs make amino acids and which plant organs put them to use. Now, the main site of amino acid biosynthesis is in the leaf. And so what happens then is that plants take up ammonium and nitrate from the soil. And these are inorganic sources of nitrogen. That happens in the roots. And then the ammonium and the nitrate are transported to the leaves through the xylem. Okay, that's part of the plant's vascular system. Think of these as being analogous to blood vessels. Okay, so these chemicals are carried to the leaf, and then the nitrogen is then incorporated into amino acids. Okay, so amino acids are made in the leaf. What happens then is that they're transported back into the plant vascular system, this time into the phloem, the vasculature that conducts food throughout the plant. And then they're transported back down to the root or up to the parts of the plant that is involved in seed production to fuel those parts of the plant's growth. And so that, the words that we use to describe the relationship of the leaf compared to the root or the seed are that leaves are sources of amino acid synthesis, and then the roots, and particularly the seeds, are so-called sinks. They receive the amino acids that are made in the leaf, put them to use for growth. Any questions about that? Okay, so the name of the gene that you guys are going to be doing all of your experiments on is APT2. And so that's a nickname for amino acid permease 2. So what this APT2 gene does is encode a protein that resides in the plant plasma membrane, just like shown here. So what APT2 does is actually plays a role in pumping amino acids out of the cell towards the phloem, where they can be taken up and transported to other parts of the plant. Okay, so it's a transporter then that in a normal plant is involved in moving amino acids from the leaf to the roots and the seeds. That seems to be what its normal function is. Any questions about that? Just think of it as a pump. Helps amino acids get from one part of the plant to another part of the plant. So this slide sums up pretty much what I've told you. Again, think of the APT2 protein as a plant, as a pump that in a normal plant is active in the vicinity of the phloem and helps amino acids get from where they're made to where they're needed for growth. Question? So you hit on the really key question that I'm building up to, which is whether there are additional side effects in a plant where the APT2 gene has been inactivated. That's a perfectly logical thing to presume, right? Because I've been spending all this time telling you about what an important gene the APT2, what an important protein the APT2 gene encodes. So the short answer to the question is that we don't know that. And that's actually part of what you guys are going to be helping us figure out.
With the inactivation of this A2 and phosphorus and limit the amount of nitrogen and carbon that they become that much.
Yes, right. That's a really good question. As it turns out, the people that have worked intensively on APT2 function in normal plants actually see kind of the opposite effects. The seeds seem to have more nitrogen and more carbon than the wildlife plants do. That's kind of a paradox, right? You would expect exactly the opposite. But But what they think is going on is that when this gene is inactivated, there's some kind of compensation mechanism that senses this problem and kicks in and actually overcompensates a little bit for the absence of that particular bump. Now, the details of how that works aren't clear, but what they do know is that in the APT2 mutant, genes that encode different amino acid pumps are activated to a higher level. So that's a great question because it illustrates the potential complexity of what's going on in this mutant and underscores why we need some help in figuring out what the consequences of this mutation might actually be. Any other questions? These have been terrific. In the back. No, not at all. I would be very interested in experiments that do involve nitrogen because I think probably a lot of the experiments that you have in mind actually haven't been done yet. Very little secret about scientists like me is that it's really easy to think of good experiments to do, but really hard to come up with the time and the people to actually make them happen. So that, one of the reasons that I love this sort of collaboration is that we can kind of crowdsource some of the things that we're really interested in that don't have the time to do. And so then that segues into the last few things that I want to talk to you guys about. So, all right, well, what are we actually doing with this mutant and this gene in my lab? We have a grant from the National Science Foundation to learn more about the function of APT2 and a few other amino acid transporters that are similar to APT2. And so what we're really focusing on now is asking whether the pathogen is reprogramming the gene for its own purposes. And as part of that, we're looking at the activity of the gene itself. In a normal plant, the APT2 gene is not active in the cells that the pathogen best likes to be. But one of our hypotheses is that, okay, the pathogen has figured out some way of activating the APT2 gene in the cells that accommodate these structures. And so we're testing that hypothesis now by making transgenic plants that have a visual marker that will enable us to see exactly where the gene is active and to ask, okay, if the pathogen is in that particular cell, is the gene on? And so we've also engineered plants with a different visual marker that will enable us to ask whether the APT2 protein localizes to the regions around those feeding structures that I was showing in that picture a little while ago. And so then, if the answer to those two questions is yes, and we think it will be, then in the long term, we want to understand more about how the pathogens actually accomplish it. Now, this is all very much basic research without any sort of short-term application. We just want to understand what the pathogen is doing to promote its own interests. But we also have some ambitions for taking advantage of this APT2 mutation in the short term as a potential tool to reduce losses to disease. Now, I was showing you guys that result a few minutes ago about how plants that have a mutation in the APT2 gene are less sick. So what that suggests is a possibility of, okay, maybe if we take the APT2 gene and something like those potatoes that I was showing earlier and inactivate that particular gene, maybe then those potatoes would be less susceptible to disease that's caused by that Phytophthora pathogen. Right? I mean, it's possible in principle from what we know now. Yeah. Yeah, that's an aspect that we're not sure about yet. It could be that they're different relatives of that gene that have maybe a different protein sequence but have the same function. But that's something that we can sort out pretty easily. But that's a great question.
Is there any way for BRVGS
Yeah, we can do those sorts of things. Our hypothesis is that the pathogen is actually sending a protein to the inside of plant cells whose job it is to bind to the APT2 gene and increase its activity. And there are assays that will enable us to detect that if that's what's going on. Exactly, yeah. Well, what it actually does is, what we think it might do is to bind directly to the DNA around the gene and then recruit plant proteins whose job it would be to make that gene more active. But that's all speculation. We don't know any of that now, but those are the sorts of experiments that we do in the future. But, you know, in terms of short-term application, we're really interested in asking whether a simple mutation of the APT2 gene itself might be a good tool for more resistant crops. But that then leads to the question that came up earlier, which was right at the heart of this, which is whether there are side effects of this APT mutation that might affect crops' ability to produce whatever kind of food it produces. And so then where we would really like to get observations from you guys are around these questions of, okay, is plant development altered? These are Arabidopsis plants that are lacking the APT2 gene. Do they make fewer seed? Do the seed weigh less? Are the leaves stunted or malformed? And then also in terms of your environmental variables, we're really going to be interested to see whether the APT2 mutant plants are more susceptible to the things that you guys might tweak in your experimental treatment. If the answer to that question is yes, then maybe that's bad news for the prospect of the gene's ability, of the mutant's ability to provide resistance. It's actually useful in field context. But, you know, the overall point is that, you know, to get at this question, the experiments that you guys are going to carry out, I think, are going to be really valuable to us in terms of evaluating whether it's feasible to move forward with a simple strategy that involves a directed mutation of APT2 for disease resistance. So I'm really looking forward to hearing about your guys' results in November. Okay, so this kind of sums it all up. Hopefully I've convinced you guys that plant diseases are important on a global scale. And I've told you that in a normal plant, APT2 plays an important role in amino acid transport, but then also has the secondary role that works for the pathogen's benefit. And so we think that a simple mutation in APT2 might be a good weapon for reducing disease, but we need to know more about what the undesirable side effects of that mutation might be. That's where your guys' work is really going to help us out. So that's all I had to say. I'd like to take more questions. There's a question here in the back. Yeah, that's a really good question. So pathogens like HPA have kind of a delicate balancing act because they require the plant to remain alive until it completes its life cycle. So they don't take everything. They leave something for the plant. But they take enough, there's definitely a growth penalty. Now for pathogens like Phytophthora infestans, they take everything. And indeed, at the end of their life cycle, they actually kill the plant and then feed off what remains. So it's different for different pathogens. Uh-huh. That's a terrific question. We're actually setting up experimental systems now to ask exactly what is the effect on resistance or susceptibility when we alter nutrient input in the plant. I want to write a $10 million grant proposal in the next few months that's meant to get it exactly that way. Any other questions? Thanks for your attention, guys.
We're going to be working on, kind of talking with them about the project. Love to have to figure out we've got lunch.
Oh, yeah, of course.
Yeah. And then they'll be talking about variables too. So look, get your input and feedback. Hey guys, we're going to take about a 1 minute stand up break. Just kind of stand up, shake out, move around for a minute. We'll get set up.
That was good. I think they got. I mean, they know the purpose really well. I think. So
Yeah,
that was good.
They asked really good questions.
I was impressed with the
Yes,
questions.
they did. My students did. Where are all their good questions? But
You'll be busting them.
I will. Probably publicly here in the next few minutes. So we're going to go over a little bit of their project parameters, and then we were saying break up into groups, start talking about potential variables, and it's like they go back from like group to group, just kind of hear what they're bouncing around.
That'd be
If you have any
awesome.
input,
Yeah,
awesome. Yeah, that's
I've also brought all of the kit.
Awesome.
So I'll bring
Awesome.
those in around lunchtime.
We've got plenty of helpful labor here.
Yes, we do. Yes, we do.
Right.
So we'll make that happen as well. So yeah, really appreciate you making time for this. That was great. These kids, they are. And they're, it's an application for the program, and they are supposed to want to be a long way to be. But it's funny, we're doing a few different things like this. We'll get them connected to higher ed, and they're kind of like, oh wow, this is what I got to be ready for. Wow, that's
Why?
and it's a great, absolutely great presentation. On that level, I think I got to really pay attention.
That's what you're not excited about. Don't worry about that. Because the nice thing is, the only part that really requires like research and like that is your intro. It's 2 pages, 500 words. Like that's it. Like one single spaced page, 12-point font, it's about 500 words. So that's it. After that, it's like materials and methods. We did it real easy to write. Our results, you guys collected them. That's real easy to do. Our analysis conclusion, that's harder, but since you've been working with these things for weeks and you've got a group you're working with, that ends up being pretty too. So the nice thing is it's a long report, but since it's in like chunks, it ends up being, I think you're going to find it, you're going to like at the end, be like, this is this behemoth thing I wrote, and this is really easy. So if that's the part that's not that you're working, don't worry about that at all. Because I mean, the thing is, you just got all the information you need, with the exception of whatever we choose to test in our class for your whole intro. Like done. Like so, we'll talk more about that tomorrow in class. But that part's going to be easy, I think.
Most of our projects do.
So about a third of the class would be up here talking about, hey, we thought we want to test this variable because we thought it would tie in this way, and here's what we thought was going to happen. Here's our data. You can see that, we were correct. What we thought was going to happen actually did. Why would this matter? Well, this thing has to do with the protein. You don't even talk about that. There'll be a poster group that's going to be kind of a poster representation of that same sort of thing. And then the other group will have already put together the website, the data, and everything else beforehand. So, and on where we collect our data.
So it's like kind of
we've gone a long way.
Less informative, more like here's like a one-stop spot if you need to see what we've been doing. It's more like not technical, it's more boring to be honest. It's like here's a graph, here's where we're collecting all our data, so you don't got to worry that someone who has the notebook left it at home would put it on the website. So it's a way for other schools to check and say, oh look, wow, look how much faster there's like growing in ours and stuff like that. So, but I think you guys will. I don't know. I'm really excited because it's like, there's a lot of technical detail you guys just got. Like the idea that you have like this fungus that blasts its way through plant cells and like sucks out its nutrients, and here's this protein that maybe can stop that. Like, it's pretty cool. So I'm excited about that. I'm gonna have you guys rank them 1, 2, 3, and I'll try to give everybody their first choice. And if not, I've only had one time where I had to kind of ask someone, hey, we need one more person in this group. I know, like, the 3 of you said that was your last pick, but it's kind of like, that only happened one class ever. Almost everybody gets their first or second pick. So, and if people are like, I really don't mind between these 2, but definitely not this one, I've never had to give someone the definitely not this one. The only time someone had to take their last choice, they were like, I don't really care which one I do. So that was fine. So if you really don't want to be presenting, that's not a problem. If you really do want to be presenting, then that's good, you know?
I'm actually not sure because I mean, I understand that it's like going ahead and
Scale it up.
out
Well, the rest
how
of us
like
still.
back to this meeting.
This is the biggest
First
class.
presentation is not
This was the biggest class until this year. Now we're up to.
I don't like things like doing that. It's
The
kind of
thing
like
is, the more you do it though, people are worried that they're going to mess up and get embarrassed. And after a while, you'll just make mistakes. That always happens. But you'll realize it like takes that experience to realize that oh, I didn't even care that I made a mistake. I didn't even notice that I made a mistake. Sometimes you guys get so caught up on like mispronouncing a word or a phrase. It's just like no one cares. People mispronounce words constantly. I'm like, I make more mistakes in one class period than you guys would make in an entire year of entire high school career of presenting. You know what I mean? I'll make more mistakes in one day than you guys make in 4 years. It's just you just don't care. I was like, whatever. I always say, imagine you're just talking to someone about something you know, like you're doing right now. That's all you do.
We're good?
Yeah, we're ready.
All right, ladies and gentlemen. Next seat, please.
What's up? We'll talk a little bit about your project. That was awesome. I put my hand up and I say shush when you guys all get quiet. So anyway, first off, me and Mr. Caraway were already mentioning this. The questions you guys asked were awesome. I know they were definitely concentrated more than others in some classes, more than others. I had here Orange County questions. So anyway, those like awesome job, really. Like pay attention, asking good questions. What I'm going to do right now is just talk briefly about what you guys are going to be doing first with your project. I'm going to get you with the project website, very similar to what you guys did in 9th grade. You don't really need to take notes for anything I'm saying here. And you'll be hearing a bunch of this stuff from your teachers as well. After I'm done going through this, we're going to start breaking up into class groups so we can start talking about what variables we might want to be testing in our various classes. And we'll be using the laptops to do a little bit of research on that. See if we can't leave today with at least an idea for what we want to use. Maybe don't have an exact like pinpointed variable, but at least a couple good choices. So anyway, if you have ever used the Governor's School website, which you probably have, you can go to academics and then current projects, and right on there is the AP Biology Arabidopsis research. I also was messing around with this because I am too lazy to go to there and click a couple times. I always just type in BRVGS Arabidopsis, and I was playing around with this and spelling Arabidopsis like Arabidopsis and Arbidops and all these things. It always came up first. So that's how I find it. So let me go through a little bit on what you guys are going to be doing. So you should have already done some sort of pre-kickoff here project, at least to familiarize yourself with the plant a little bit more. You can see on here we have a countdown to when your research papers are, presentation day, everything like that. Again, the before you begin part is stuff that we've pretty much already completed. But what I want to talk about right now is the, whoops, not the experiment itself. We're going to be getting into that here, but the research paper that you're going to be writing. On here is a little overview of every single part of this paper. And I told this to my classes. I always try to, not on purpose, intimidate people, but kind of on purpose. And I'm like, you're about to be writing maybe a 10 to 15 page paper for this project, and people freak out, but you will be surprised how easy it is to do this. You'll have this massive report when you're done, and it's actually pretty simple to write. The only thing that you need to really be worrying about right now is the introduction. If you were paying attention and taking good notes, got almost all the information you need to complete this background research part. The only kind of missing piece right now is the variable that your class is going to be studying. What effects does that variable normally have on plants, specifically Arabidopsis, if you can find that information. Again, the idea here is that the effects that it has on more complicated plants may be the same in Arabidopsis. So what variable, the effects it has, your hypotheses, what do we expect to see maybe with our experiments? That's the one piece that's missing that we're going to start working on today. Everything else for your intro, the significance of the organism used, the reason this research, why do we care about it in the first place, what's the difference between the wild type, the normal of control, and the mutant plant, all that stuff, you just got all that information. So again, hopefully you were paying attention. That's the first thing. Technically, you go home tonight, if you knew your variable, write your entire introduction. It's the only part of this that really requires a lot of extra research. So anyway, your teachers will be talking to you a little bit more on that. The rest of it, again, we'll be going over in class. Just so you know, because this is really frustrating for us, all the rubrics you need on how you'll be graded for every step of this project, they're on here. People are always like, I don't understand why I missed points on this. It was right there on the rubric. It was right there. You just didn't do it. So of course you lost points. So don't be surprised if you ask your teacher a question and they say, did you check the project website? And if you didn't, get ready to check the project website, because that's probably what they're going to tell you. Just really quickly, what you're going to be doing from here on out is we'll be collecting data in the classroom, working as a whole group, and you may have already been given your smaller groups that you'll be working with in class. So again, collecting that data, we're going to have a website for every class, all our data that other schools can look at. We make that private within Governor's School, not open to the public, but you'll be able to see what other schools are doing. They'll eventually, 55 days from now, when it's presentation day, some of you will be up here presenting. It's not the type of thing where your whole class gets up and you're all up here presenting. Some of you will be explaining the findings you found. We have scientific posters we'll be making. Some of you will be in that group. Some of you may have been more the nuts and bolts behind the scenes people who were doing all the data and number crunching before that day. And then on that day, we usually have that group go around and ask the other poster groups questions based off of their research. So start thinking about what group you might want to be working in. Teachers will let you know when they're going to decide that. Just a good rule of thumb. If you are like, don't want to be in the presentation group, you should probably put it there, because you never get better at presenting unless you just keep doing it. So anyway, that's what's coming up with your project. Again, familiarize yourself with this site. It's your best friend for this project, especially on what you need to be doing for various parts of your paper, which is this first part that you need to be worrying about now. Other teachers, anything else we want to add? Ms. Dexter, you always have something to add. Yeah. And you can see here how that gets broken down. The biggest part of your grade is your own. The report, you certainly collaborate with your class and your group, but it's your own writing, it's your own report. If you are thinking right now, but wait, I wasn't in the poster group, but the poster counts for 20% of my grade. Correct. Though your whole class will contribute to your grade. You're not working with other counties, so you can't say, oh, I got a bad grade because some kid from Madison didn't do their work. Say that. You don't get to say that. You don't get to say, well, that idiot from Orange never answered his email. You don't get to complain about that. You're all responsible for each other here. And so hopefully that's good. And that's not like a scary thing I just said. Anyway, that's something worth keeping in mind. Your grade, again, is a group process, so keep that in mind. One person who's sitting over there in the corner and has no idea what you're doing every day in class, that person's contributing. He's contributing to your grade. So he raised his hand. He volunteered. So anyway, just keep that in mind, okay?
Help out with group. It doesn't mean because you were assigned to a group that you aren't helping, because at the end of the
So again, hopefully that's not a terrifying statement. So anyway, do you guys have any questions at all on sort of what you're going to be accountable for and things like that? Go ahead. For the whole paper, so like just real brief, like kind of nutshell overview. You have an abstract at the beginning. It's usually 150 words or less. Intro ends up being about 2 pages, about 500 words. Like you have a bunch of your materials and methods, maybe another page or so, about another 250. Keeping track with your math here. You have a bunch of data. You have some descriptions of that, and then you have your analysis conclusion, which ends up being about 2 pages. That's where all this stuff is data. And again, one thing I'll give you just a little bit of advice. Piece of paper, but chunk. What's the word count for the whole paper? Just think about it like, what's the word count that I need to worry about for this section? And you guys probably won't have issues with the word count. That's more just a way of saying, make sure you're thoroughly explaining yourself. Here's a minimum sort of amount that we think you would need to do that. So don't think of the word counts as super restrictive. They're more just, you probably can't say everything you need to if you're not saying this much at least. Question? Go ahead. Probably not. It's your paper. They didn't study. Would you have access to past research papers? I mean, they were on a different mutant. They were testing different things. This is your own. I mean, it's not like you couldn't see it, but I mean, we have a research paper about this particular mutant available to you. But past student ones, at least for me, I'm not going to let you see them. So, anything else? Go ahead.
So I do want to point this out. So you're writing your introduction first. Now in the paper, when you present your paper, upload your paper to turn in abstract. When do you write your abstract? At the very end.
Yeah, it's a summary of everything.
An APA.
Yeah, the word citation, that means that, you've actually cited something somewhere in your paper. It doesn't mean you wrote a list of links at the end. So that's a problem we always have. Go ahead.
If you were looking for the minimum amount of words you should have, it's 2,400.
So that's about, so 2,400 ends up being about 9 pages or so. So maybe more than that. Math is hard. 10 pages? What's my problem? So about 10 pages. Any other questions? Someone else had a hand up over here. Shh. Anything else? All right. So anything else from other teachers? Yeah, I'd say at this point, what we're going to do is, so don't get up and start moving around yet, please. So we're going to start breaking off into various groups here. You're already seated by class. We don't have to necessarily stay right in these seats, but you guys can see we don't have a ton of room to move around, but we can make use of the space up here and some of the other space. So what's up? Oh yeah, stop talking. Seriously, you're gonna stand up in a sec. You don't need to talk about it. So anyway, right now you need to find your teacher. You're gonna be meeting up with them. We're gonna send how many people?
Send 3 people per class. So like Orange and Goochland, get to your actual blocks, and only 3 people come up to get computers. And you'll have to tell me which class you're with.
All right, so at this point, if we could break out into our groups, and again, we need 3 people from each class coming up to see these out. So first block and second block. You guys want to come up here? No, the groups don't matter, just your class.
Watch the board. Watch the board.
Let me.
That's what you break the news.
What did you say? Let's go around and make sure.
I will tell mine.
I don't know.
I'll go. Right, folks. So right now, block one is come over here, right? Chair, whatever. Block two is this area. So, did you guys get your computers yet? Alright, so let's see if you can get those laptops. And again, if you want to grab your chairs, bring them up here, grab some close ones, that's okay. Guys, this is somewhere where I can talk to all of you. Here, I would say about the time you go today, we'll have right now. We're just going to expose a totally so what I want to say is let's get down to these things, pull that thing down on like some sort of environmental stress, something we can do to maybe see how these plants are affected. But the whole class will be tested in one area. That's what I'm saying. You've got something that you're particularly interested in, and that's good if you got kind of self-test class. So anyway, you have the computers, let's do this stuff. What effects do they have? How would we be able to tell what the effect is? So anyway, it's pretty loud, but again, let's make sure those voices can be heard here. You guys can start doing that. And again, if you need some chairs, we got them over here. So you're very good out. We're not no. All right, guys. So, the little gauntlet of people we've got here. Here's what we need to figure out. I was saying, figure out what causes one source of environmental stress, one variable that we experiments to. All of those things we determine. Stress to see did the inactive protein pump did it respond in any different way to this stress? And we're listening to Dr. Falcon say, so what are some things we can natural setting. So it can't just be like we're gonna shop in the electricity or something. It's something that we'll plan on, something that might be a natural setting. Sure, I mean, like trout is a good one. That's certainly a good one to test. And if you would listen to that, so get someone in here, what might you want to test? Figuring out now or less than that is okay. But and so the reason we measure is sort of figuring out what we would expect to get out of treatments. Stuff. No, good question. This is for the whole class. All of us are going to be using the same variables in the class, not like do this. Everybody's going to test the same variable. You don't have to. If you think that would be easier than one big group thing, if you know who I already treated, but I can break out into that, that's fine. If you want to get the three groups all themselves for the computer, that'd be okay. However you guys want to do it, but then I'll go back and talk to you and see what you came up with. So,
We have to have this
Go
variable.
out that way.
Oh, 2 rules. It has to be something that can't kill the plant straight up. It has to be something that the plant would be exposed to. I would say, like, this would be like a natural thing. Be like a pollutant. But it needs to be something it would actually encounter in its natural. So, not like, hook up an electrode to it and stop it. Or if you were going to test the acid, you want to test something like sulfuric acid, because that's a component of acid rain. Like something natural and something that just straight up won't, like any sort of radiation the plant it's not coming back. So something that won't just straight up kill the plant. Something that you guys want to think of here is, first off, you're all going to be, either getting in smaller groups in your class, you're all going to be testing one variable. And it's kind of hard to take like every single thing that could possibly affect the plant's growth and figure out what you might want to test. So here's kind of what you should be thinking about. There's a variable you pick, has to be something that just won't straight up kill the plant because you have no data. Like we found out that radiation killed the plant. Like great, like but that was kind of a crappy experiment. And it needs to be something the plant would actually encounter in its natural environment. That can be a human pollutant or something like that, fertilizer, whatever, but it needs to be something that you would actually encounter, not like we're gonna pour Gatorade on the plant and see what happens. Okay, so something natural, something that's not 100% fatal. To kind of help you out a little bit, if you're paying attention, which I know you were, is and I didn't mean it sarcastically. Like Dr. McDowell talks a lot about how this gene affects nutrient kind of movement as well as seed production in plants, but also that we don't know how this gene might affect the rest of the plant's normal growth. So honestly, any variable you can think of that will the plant's ability to handle nutrients, the plant's ability to produce seeds. Now it gets you like pH. How does that affect the plant's ability to produce seeds? I have no idea. Like, I honestly don't know. So anything that might affect seed production is probably a decent place to start. So good.
Hey guys, as you probably noticed, the internet has slowed to a crawl. So if we can have one person in each class just shut down their computer, so we just have 2 per class, and see if Just Western can handle us then.
So that's really good. We've always set, like when we plant these seeds, we've always set their soil to a very particular level. I don't know if we've ever tried something where it's actually less, so you're kind of physically giving your roots less space to grow. That's pretty
If you would like to use a cell phone to do research, you can take it out as long as you're using it for research.
I would ask Dr. McDowell maybe like write that one down. Like just think, you would come up with maybe a list of 3 things that okay, we'd be okay testing this, but we don't think they're the most boring thing in the world. Like don't pick a variable that you like. We're gonna test this copper compound. No, don't do that. Pick something that, okay, this is a component of fertilizer. We know it affects plant growth. Something that you can find lots of information about and like apply it to this new plant. So when we leave here today, you want to have a list of about, like I said, 3 things. We'll email as teachers and kind of say, here's our list. Okay, let's figure out what we're going to do. So if you have one that you're like, for sure, we want to do this, no problem. Most of the classes don't really come up. They have like a couple things they want to try. Again, it's not your research. It's kind of hard to be like, what am I actually interested in? Because it's like the problem's being kind of forced on you. You need to figure out how you're going to test it. So think of something that, again, I want to say think of something easy, but think of something simple versus really complicated. Think about how would we actually do this. So lowering it in the soil, that's pretty easy to do. Adding chemicals to the water, that's pretty easy to do. Changing the frequency and the wavelength of light that's reaching it by like putting a filter over the lens, that's really hard to do. So it's like temperature-based things. That can be easy. So it's like, think about that type of stuff. Competition. The only thing I'd say is bad about like competition is you've got a lot of aspects that are potentially effective. But what you could do is the idea of overcrowding. What you can do is plant these seeds. You can only really have about 6 plants in a pot, or else they're just going to leach materials off each other and not going to grow very well. So you could have pots where you need more competition rather than definitely test this. That's definitely going to affect the plant's growth. Maybe these newer plants that can't be moving amino acids easier will have a much harder time with the overcrowding. I don't know, but like that's definitely a good one. So that's kind of what you want to think about. Anything that affects plant growth, anything that affects seed production, go with that, and maybe that'll help lead you to something a little bit easier. Oh yeah, you got it.
What about when you got like a fungus or something?
That make it a little bit easier? You guys look swollen.
So, what I would say to them is, Dr. South mentioned, don't, I think this is so cool. And I would say for you, because it's hard, given the situation where it's not like I know the answer, but you're not going to see that I see the reality. Yeah, they know infect this protein release disabled, affects the way they know that it affects the growth of this fungus. They don't really know the other effects it has, which is why we are going to try to test stuff. So like I said, getting the nutrient related, getting both related, think about how we test it. If we're modifying the soil, modifying something with the water, if we're modifying if it's not modified with lower light, that starts getting really difficult. Should I soft light set up? How would we cover it up? How would we control the temperature? That's just really hard. Again, I don't think question eight. So make sure that you guys are coming up. Think about how we would actually do it. You guys have one that's kind of like top 10, top 2, top 3. So would we be testing temperature? Would we be testing pressure? Something we're looking at is how does seed production or plant seed production in general? How does that affect? What is the normal range of temperature for Arabidopsis? Are they changing or not? What we do is Arabidopsis is cold drainage. We like less than surface temperature. That is something we can do. We can't like put things in the fridge play around with stuff like that. That is possible. So what else would just let me know the specific type. I will say one problem is try to add to the water. What is the quality is you're using like osmosis breeding ground and like mold and stuff to grow in the soil, that's happening. That's usually one that we try to avoid sugar-based stuff. Because like you're fighting straight up nutrients for everything else in the plant that's what we might want to be careful about. Not saying we can't do it, but we may want to ask. Just because we drink water, it's very easy to modify. I'm not saying you should pick that. I'm saying those are the water. Okay, so what I would say is, look up chemicals. So we're going to have a normal wild type control. And you're going to have and it's had that team. We're going to compare those two and say, what is just a very superficial level? How do they grow? Can we see any difference in species? I'll show you one example of the variable. Does the mutant respond in a different way than the true mutant? So what's just an keep thinking, guys. Narrow it down a little bit. Let me keep reading it. Dead plants. Yes. So when you try to diversify that, it's hard to do on the If you're doing excessive nutrient, what's the nutrient? You know? Oh, what'd you say? another different time. Anything growing so that is actually things that people have tested in the past, although it's hard to control. But they've tested things like the effects of predators and stuff, and you by carrying leaves and stuff and see how the plant responds to that. Without actually damaging the plant, something I was telling them the same thing. What you might want to look up is if a plant is damaged, what does it do to respond? What is there something we can test that would actually like, oh, this is something that a plant releases whenever it's very damaged. I don't know what that would be, but find it, we can test it. This is awesome. Like, I think these are great, and they're not difficult to do. So Well, I was saying like maybe fewer seeds, which makes sense because there's not as much nutrients to kind of do it. Like reducing takes a lot of extra nutrients that the organism usually isn't having. But I'm like, I can also see conditions from bad. Maybe a plant focuses more of its energy on reproduction because it's like I am done growing because these conditions are bad. So let's just produce as many seeds as possible. Do you know what I mean? Like I can see both. I actually don't know.
All right.
What are we thinking so far?
Oh, we've got everything from diesel fuel to keratin. How about you, Bruce?
Mine are keeping things somewhat traditional, which is these would be like maybe drought. That's a good one. Sugar-based stuff.
And mold.
Bacteria and everything else.
You guys seen the professor's cat?
No, I have not.
I didn't know.
You know, he was here.
The girl in the green. Yes.
We've already talked about it.
Yeah, so did one like it.
Here's what I said. Okay.
Like best nature thing is why I said.
How many different variables?
Right, and it's just a cool idea.
It's either acceptable or not.
So, how many trips have you been on for this? Yeah, for the question.
In the past?
Yeah.
2.
2. Okay, thank you.
Yep. I've been on 2 in the past.
So like probably about 4 years, so yeah. Sure. Yeah. That's fine. Thank you for doing So I guess what we'll do after today is kind of, I mean, we can even have them sort of say, here's what we're kind of thinking right now, and just hear it real quick, and then the teachers can sort of figure out. I don't like when they test the same thing.
I agree.
In the schools.
And if we can sway them to do something different,
Right.
then
So
your
we
group.
could say, like, here are the top maybe 2 or 3 things we're thinking about right now. We can even make them say, like, come up real quick and say, here's what we're thinking. We don't have to do that. But
That's what I'm going to make. I'm going to whole group here in just a second and say, whittle it down to your top 3 or 4.
right.
So I'm good with that.
So say that. So if we had to have a representative soon.
You know the time or no?
I would say, I think Mr. Kelly said like 12:15. So that's one thing. Relax. Here's what we're thinking for our variable. Top 3 things, just so everybody's hearing what other classes are doing. And they say, okay, well, back to the we're going to do this. So pick your first one in mind. Just say that. I mean, just maybe like what's your justification for why you're doing it. You know, if you can't justify why you're picking this particular variable, then it's probably not good one. But I think you guys are way past that. You need to think that. Hey guys, if any of you have like a scrap piece of paper or anything, you could just tell me sort of like real quick top, right anywhere you you care about, you can like let's stop it at 3. I know you guys probably don't have that many, or maybe you might, but like you got like a top 3, top 2. Tell me what they are. And just real quick blurb on why you want to test that. I'm gonna bring that back to the other teachers, and then we're gonna talk about that real quick. We'll make you guys stand up and say it. So. If you guys are done, you can bring back Ms. Alton. What's up? What you got? So how did they the thing I'm worried about again is the bacteria in soil just because we have evidence that is not happening. If we can figure out a way of doing this in a concentration that's not going to cause that problem, then I'll be able to create directly applies to seeds, which is really it seems like which because at the most basics, like we said, 50 to 99% of these share a chunk of our DNA. That's the idea. You learn how to it's basic and you figure out how to apply it. Ellie, did you have it? Or good, whatever you have.
Anything we can or should we kind of winding down?
Right, I think so.
But we want to know it's about 3 minutes to 12. So the part of me that absolutely hates this is just on purpose students is wondering what purpose we might give them in their young lives between now and 12 and 15.
So what we were saying is what we want for these classes to do is kind of have a representative something sort of let's report out to the group at large. Here are the 2 or 3 things that maybe we want to focus on. So that way if like if Louisa says we want to do this thing, here's our kind of top choice in my class. It's like, well, that was one of ours, but we had this other thing. Like so we're not overlapping because I'd rather see different things.
Right.
So I think kind of do that here, and that would be kind of the start.
So my sense would be to start to tell everybody we're going to pull it in the next couple of minutes.
Right.
And start doing
And I would say we have a way of randomizing the order, and then you have that group report, and then that could be the first thing. So,
right. Okay.
Like you're right. Like
here, I'll give you a randomizer. I got your randomizer.
Oh, of course. So I have a question. So one young man asked, do we know, you know, how the pathogen you spoke of at the beginning, how it circumvents that century? Right. So that's a really good question. It's really complicated.
We know some probably doing a lot of different things at once, but most contagious strategy is send in, that's what our pilot model does, because he gets in the before I come up with the
Oh, there's an invader. Yeah. Right. Yeah. He's just thinking down that line. I like it. Oh no, it's a really important
I got to start getting chairs back in rows. There's a couple things to do before you
Now, on the prep website, there's very nice documented Right. Yeah, okay. So, the first hypothesis
right, ladies and gentlemen, and all you people with phones up, phones up. I appreciate that. That's Mr. Caraway. All right. All right, a little bit different order of operations today. Usually we'll have lunch and then finish out with some different activities. But today, what we did was put lunch at the end of the day because we had all this kind of crazy stuff to get done. So it's going to be here in a few minutes. It is coming. We got a lot of questions about lunch. We are going to have lunch. That's going to be the last thing we do today. Before we get into that, one of the things we wanted to do was, you guys have been making decisions as individual classes about the variables that you're going to apply to this experiment. I think what we want to do is hear from, is to have everybody hear from everybody else, essentially, and then some discussion on that. So, Mr. B, I'm going to turn that over to you.
All right. So, first off, it's sometimes hard because it's not your research interest or anything like that. I mean, you're taking the research from somebody else and furthering it. So, good job with you guys coming up with stuff that is feasible. First off, not just something kind of silly like I always say we're gonna put Mountain Dew in the plants. Like I didn't hear any of that type of stuff going around, so that's good. What we want to hear right now is and me and Mr. Carroll used a highly sophisticated randomizing technique to figure out what schools would be going first here. We want to hear from a representative or two from each school. Kind of stand up. Let's hear what you guys are thinking as far as maybe first, second, third choice on what you might want to be testing. The rest of you out here, as these people stand up and say, here's what we might want to test, compare that with what you were coming up with. If their first choice is your third choice, well, like, let's start whittling it down. So, because there's a lot of overlap with what we've been hearing walking around the room. So, again, we just want to hear, what are you thinking right now? If you have a reasoning for why you're picking that so far, that's kind of what we want to hear. So, lucky, Nelson, you're first. So, quiet, folks.
We wanted to test the effects of overcrowding.
And so something to keep in mind too is when you have, we say like fertilizer, you guys don't have to keep standing up, but it's like fertilizer, something like that. Just keep in mind, like you want to maybe find a particular component, particular nutrient, something like that. And if anything to contribute to, feel free. All right. So orange block 2. Let's hear it.
So we stole my sheet of paper that I had everything nicely written down on. Thank you. So our first choice was excessive nutrients and C, but like we kind of narrowed it down. We picked 3 just today though: calcium, phosphorus, and magnesium. And then for our second choice, we had drought, seeing what would happen when you take the water supply. So,
yeah, I want it back. Thank you. All right, folks from green, what do you got? So why heavy metals? So common source of pollution for sure. So, so your first 2 choices, like those 2 metals, your first 2 choices. Okay. Do you guys have anything out of the heavy metals or? Oh, that's fine. I just was curious. So I didn't know if that was one thing or like 2 separate things. So that's good. So this is actually good so far because I haven't been like, you may have talked about these types of things among your group, but we haven't seen like a huge overlap yet, which is always good. It's always cool on presentation day when you're like, this group tested something completely different from mine and had completely different effects. So that's always good to see. Madison, how about you?
So at first, we kind of discussed what excess amounts of carbon do for the plant, and we also discussed what hydrogen peroxide would do, but we're really going to lean towards what would is going to do for the plant, because that is the most present amino acid in wild type groundnut.
That's awesome justification for sure. Goochland, I don't know what blocks your classes are. We wrote Goochland 1, so I guess that's 3.
We looked into overnutrition with nitrogen and adding excess nitrogen. And our second choice was chronic stress, like.
All right.
Now, I want to back up because we had talked about nitrogen, but they talked to Professor McDowell about that, and it was very specific reasons.
He was doing nitrogen water, and he wanted us to compare his results to ours.
Sure. I think that's good.
He said his results were being done in water, growing plant water, and that would be interesting what the results would be if we did ours as well as compared to.
How about, so, I can't, where was I? Orange, block 1. What do you got?
Okay, so the first one we were doing was higher levels of salinity in
introduction of seeds. So we can see how that affects the plant, and then.
Okay, so we were doing cell wall growth. Then John and I narrowed it down to sugars. So glucose is made by photosynthesis, and that is then turned into sucrose. So sucrase glucose too. That takes a lot of energy out of the plant, and since the pump is turned off with all the protein getting into the plant, we were wondering if we could decrease the amount of the enzyme that breaks down the glucose. Because the reason why it's turned into sucrose is because it survives the phloem easier to go throughout the plant. So we were wondering if we could decrease the enzyme, which stores the glucose, and see if we could the energy would then be produced from plants like physiology.
I don't know how the heck we would test it, but it sounds. I mean, I like it definitely. Like there was, I saw the research backing it up, but I'm like, man, how do we test that? So, how do I make this easy for me? All right, Louisa, what do you got?
So at first we were thinking about maybe doing an excess nutrient, but then we decided that a hormone might have a greater effect on it. And leptin is a hormone plant that controls the amino acid transportation. But then we realized that salicylic acid might be easier to find, and there are different effects that salicylic acid has with leptin.
All right, Fluvana.
So we actually brought it down to a top 4: calcium, manganese, not manganese, potassium, and sodium. So we chose calcium and manganese because both of those are directly related to some of the effects that the gene that we're changing does, like protein and amino acid transport and molecule assimilation.
Our reason behind potassium and sodium would be because when there's not enough potassium, sodium kicks in. They can be used interchangeably to send more proteins and energy throughout the whole plant. Also, potassium and sodium keep plant stems stronger, so we're hoping it will keep the plant upright longer, because when the seed pods get too big, the plant tends to leave. So that's what we're hoping to see.
All right, and then Goochland block 4, or whatever it is, 4. So, how would you? Did you guys talk about how you would test the gravitropism? I'm just curious. I'm not trying to like call you out on it.
We decided that we looked up exactly what some examples were in nature, and the example was a tree actually growing up and down.
Mm-hmm.
And we looked at the descriptions, and we were thinking about just placing the planters on the side, and then the ones more on top could get either more sunlight, and we just see the different effects that it would have.
That sounds good to me. So these, I don't know. We've never actually made the groups have to report back like this. Oh, you can I can tell heads are turning. So anyway, these are actually really. Teachers are going to eat all the pizza first. So. All right. So anyway, these are actually great. And the thing that I think has been best so far is that a lot of you have stated your justification for exactly why you are going to test it. Because that's something that I think, at least, that I've noticed in the past, is that a lot of times students pick variables and they don't know why they're testing it. They're just like, we just picked this thing. And they don't really tie it into the overall problem. So the amount that I heard from everybody, actually relating it to what you heard earlier, I think that was awesome. Really good stuff. The next thing you kind of need to be thinking about is how you're going to tell, whether your treatment had an effect, what type of data you want to collect. And you may have done this already, but when you're talking about expecting the plants to stay upright for a longer period of time, well, that's pretty clear on what you're going to be looking for, to tell whether that was the case or not. So the thing you want to start thinking about now and doing a little bit of research on, if you haven't seen it already, is how are you going to be able to tell that your treatment actually had an effect? What sort of qualitative, quantitative things are you going to be noticing? Are you going to be trying to count seed pods? Are you going to be just height of the plant? How coloration? What are you going to do to actually figure that out? So that's sort of the next step, something you're going to be talking a lot about in your class. we'll prep the seeds over the weekend and then actually plant those and have them ready to go next week. And then for most people, I forget 1 week, 2 weeks. I always forget how long you wait till before you start applying treatment. 2? I always forget. So I have to look it up every year. Yeah.
So when I went to my group to let them know that we were going to be sharing out what our variables were, they're like, let me taste it. So I just want to say what we just did is we were doing science. This conversation about what our ideas are about testing. This is what scientists do. Is they talk about this. It isn't some sort of contest out there. The contest then doesn't make it science. So that sharing information and thinking about what other scientists are thinking about doing, that's good science.
And like, if you thought that any of the variables that another class said were interesting, I mean, that's part of this project is you'll be checking in every other week on sort of the progress that the other schools are doing and seeing, okay, they picked this thing. Here's what they thought was going to happen. Here's what they're actually noticing. Look, you can see pictures of the plants as they go, see how they're being affected by the variables. So you will be, even though you're not working directly with the other schools, you will be still seeing what they're doing and being able to sort of interact with that data as they collect it. So anyway, are we, Mr. Caraway? Okay, so just sit tight. Hi.
Hey guys, I have one more laptop out with students somewhere.
Still have a laptop?
Who is
Okay, you guys are hungry, right?
If we're not listening, we're not going to be serving lunch.
Quiet, we can hear Mr. Caraway.
Oh, thank you. First of all, we have an announcement from Ms. Mack regarding recycling.
Yeah, it's my thing. I reduce, I reuse, and I recycle. And I'm not doing a whole lot of reduction because we got a lot